should also really be review for you. This one is all about mitosis. So hopefully you'll kind of see some things that remind you of regular biology. And basically, once we were able to develop special dyes to observe chromosomes, we were able to observe mitosis and cytokinesis in great detail under the microscope. And that's kind of how we started to figure out how this process of cell division works. So the cell cycle really has two parts to it. The first part to know about is the M phase, or the mitotic phase, and this includes mitosis and cytokinesis. And then interphase takes up the rest of the cell cycle, and this is where the cell is growing bigger. It's copying chromosomes to prepare for cell division and just doing normal cell stuff. So interphase, as you can imagine, since it's when the cell is doing normal cell stuff, takes up most of the time in the cell cycle. About 90% of the cell cycle, the cell is in interphase. So if you're looking at a whole bunch of cells under the microscope, which we will do, you're going to notice that most of the time those cells are in interphase. Interphase can be divided into three subphases. So the G1 phase is gap 1, the S phase is synthesis, and G2 is the second gap phase. So the cell is growing and getting bigger during all the phases, but especially the G1 and G2 phases. The S phase is the synthesis phase, and it's called that because that is when DNA is being synthesized. It's being replicated. And so the S phase is when the chromosomes get copied. So G1 you can kind of think of as like a brand new cell. It's growing and getting bigger just to be its normal cell self. In the S phase, the DNA gets copied, and in the G2 phase, the cell is getting even bigger because if you recall, by the end of mitosis and cytokinesis, that's going to split into two. So that's kind of how you can divide the phases in your brain. Here's a pretty common image we'll use of the cell cycle. So you can see interphase takes up most of the time. It's also in G1 for a long time. S is when the DNA gets copied, and then G2 is when it's really getting big. And then just this tiny sliver is the M phase, the mitotic phase, where we have mitosis and cytokinesis, and we get the two identical cells. And then one of those cells, each of them will actually go through the entire process again. But you can see interphase takes up most of the time. Mitosis we divide into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. And then towards the end of mitosis, we get cytokinesis. So if we start with the cell in G2, so still in interphase, you can see our chromosomes are duplicated, but they're still not condensed. They still look like spaghetti, kind of like the chromatin. You can see the nucleolus here. The nuclear envelope is still in the process is still formed, basically. It's still present. And then we have the centrosomes with the centriole pairs, and they're just stuck up here. Nothing going on. In prophase now, you can see we are beginning to get our 2 sister chromatids condensing, and they're starting to form these X's. And then as prophase is also beginning, these thin strands that we call the mitotic spindle start to emerge from the centrosomes and form this complex called an aster. So I'm actually going to skip ahead so you can see this a little more closely. So we've got our aster, the spindles are starting to form in prophase, and the chromosomes have condensed into their X-shaped form, including 2 sister chromatids held together by the centromere. In prometaphase, the nuclear envelope has really begun to dissolve and is no longer present. And additionally, the spindles are really starting to stretch out from the centrosomes and the asters. And then in metaphase, if you can recall, meta means middle, the chromosomes in this step line up in the middle at what's known as the metaphyseal plate, or the metaphase plate. That's just in the center of the cell. So you can see the sister chromatids are lined up nicely in the center, I think. This one, yeah, you can see a good picture of them here. And now you can see the spindles have reached all the way to the center and are attached to the center of the chromosomes. In anaphase, the sister chromatids are going to be pulled apart by the spindles as they start retracting back towards the centrosome. And then in telophase, we begin to get in animal cells what's known as a cleavage furrow, where we can start to see the cell division happening. But remember, mitosis is just about the division of the nucleus. So after the cells separate, or the chromosomes separate in anaphase, in telophase, the two nuclei start to reform. So those nuclear envelopes start to reform, and the chromosomes start to decondense. They kind of get loose again. They're no longer tight. And they've also been separated. So now we have an identical copy of each chromosome in each of the new cells. You can also see the spindle fibers have retracted the microtubules back into the centrosomes and the asters and are no longer visible. It should also be noted that when this slideshow got converted to Google Slides, all of these phases have E at the end. I think metaphase and anaphase, for some reason, it disappeared, but those are present here. Now, after telophase, cytokinesis is going to happen, and we'll talk more about that in a moment, but then the cytoplasm will divide and you'll have the two new cells. But once again, mitosis is just the division of the genetic material and the division of the nuclei, and then cytokinesis is when the cells actually divide. So you can see here, there's metaphase, them lining up, anaphase getting pulled apart, telophase really getting pulled apart, and cytokinesis is starting to happen here with that cleavage furrow. Now we're going to talk a little bit more about the spindle. So the spindle, those thin fibers that you saw pulling the chromosomes apart, is a structure made of microtubules and proteins, and they are really special because they're the ones that are controlling the movement of the chromosomes. And those are very important because, as you can imagine, if the chromosomes don't get pulled apart properly, you're going to have messed up DNA and you won't get those identical cells. So in animal cells, like we saw, the assembly of the spindles begins in the centrosome, which is kind of like an organizing center for those microtubules. As the centrosome replicates during interphase, you get two centrosomes that move to opposite ends of the cell during prophase and metaphase. That's when the spindle fibers move to the poles. And then the aster is that array of the short microtubules that extends from each chromosome. So the spindle technically includes the centrosomes, the spindle microtubules or spindle fibers, sometimes they're called, and the asters. Then during prometaphase, some of those spindles attach to the kinetochores of the chromosomes and then begin to move them. And the kinetochores are these protein complexes that are assembled on sections of DNA at the centromeres, and they're called kinetochores because they're what the spindles attach to help the chromosomes move. So kinetics, movement, kinetochores. And then at metaphase, the centromeres of all the chromosomes are lined up in the center at that imaginary midway point between the spindle's two poles, so that the spindles can then pull the chromosomes, or the sister chromatids rather, apart. So you can see here a little bit more closely. These are your spindles, the spindle fibers, the microtubules. Here's your chromosomes, and they're attaching to the chromosomes and moving them apart. And then this is the centrosome, which contains the asters, have all these microtubules. So then, like we said, in anaphase, those sister chromatids separate and move along the microtubules to the opposite ends of the cell. And the microtubules make that happen by shortening at their kinetochore ends. And there's also some motor proteins that we know that help bring the chromosomes to the ends of the poles. But in reality, we actually still know very little about how this process works. Scientists are still kind of trying to figure out how all of this occurs in mitosis, but it is pretty reliable every single time. Obviously, there are some errors, but if you think about how often cells divide, the percentage of error is actually very low in comparison. So they did an experiment kind of where they marked a piece of the spindle to see which part was getting shorter, and they did find that it was at the edge of the spindle fiber that was shortening. So they made this mark, and then what they observed was that it was shortening from this long end over here. And so that's where we believe all of this action is happening. Now, at the end of anaphase, we see that the duplicate chromosomes have now arrived at opposite ends of the parent cell, and then cytokinesis can begin. And cytokinesis may begin during anaphase, or it could begin during telophase, and then the spindle disassembles like we saw in telophase. So now moving on to cytokinesis, in animal cells and plant cells, cytokinesis happens differently. So in animal cells, which is what we've been looking at in all of these diagrams so far, cytokinesis occurs through a process called cleavage and forms a cleavage furrow. It's kind of like a little pinch in the cell, and then eventually it pinches off and separates. In plant cells, a cell plate forms during cytokinesis, and it's kind of just like a little divider, almost like a new wall that forms, and then you have two new cells. So in animal cells, you can see we get this cleavage furrow, and like I said, it's like a little pinch, and then the cells separate. In plant cells, this cell plate forms, and like you can see, it's a little dividing wall, and then it just forms a new cell, and you get two new daughter cells. So it's really more like you just add a wall in between the two cells where the genetic materials have been copied. You can see the two nuclei, we get the wall, two new cells, as opposed to an animal cell, which actually physically divides. So there's the animal cell, and there's the plant cell, and the cell plate is forming, you can see. This is another picture of a plant cell. So prophase, you can see the chromosomes are condensing, but we can still see the nucleus. In prometaphase, the nucleus has disappeared and the chromosomes have continued to condense. In metaphase, the chromosomes are lined up in the middle. Anaphase, they're moving away from each other. And then in telophase, we get the decondensing of the chromosomes. They're coming back to be more like spaghetti, like chromatin. The two nuclear membranes have reformed, and then here's your cell plate forming. And here's up close. Prophase, prometaphase, once again in the plant cell. Metaphase, anaphase, telophase. The last thing we're going to talk about is binary fission. So prokaryotes reproduce by a type of cell division, called binary fission. And like we talked about, a lot of times prokaryotes are single cells. And so when they divide their cell, they reproduce not only the cell, but the entire organism. So a lot of these prokaryotes also only have one chromosome. So for example, in E. coli, they have an origin of replication where that is the place where replication of the chromosome begins. And then once the entire chromosome has been copied, the plasma membrane kind of divides and you end up with two cells. So you can see here, this is our origin of replication in the E. coli. Now we have two origins of replication after the chromosome has been copied. You can see origin and origin. And then now that there's two origins of replication and all the DNA has been copied, we can pinch apart the cell and we end up with two identical daughter cells, which are also, because remember this is a single-celled bacteria, two identical organisms. So always coming back to evolution, since that's kind of the overarching theme of this biology course. Since prokaryotes evolved before eukaryotes, we believe that mitosis most likely evolved from binary fission. And one piece of evidence for this is that there are certain protists that exhibit types of cell division that kind of seem like an in-between binary fission and mitosis. So you can see here, these are like dinoflagellates and diatoms and yeast. So the dinoflagellates still have microtubules, but they also have the chromosomes that are kind of copying. And then even the yeast and the dinoflagellates, it looks even more like mitosis. So that is the end of section 9.2. And then in 9.3, we will be discussing how this process